will try to show you exercises with the different modeling platforms and softwares. So the aim of this afternoon session is that we go into a bit more into the detail of how to run these different simulations that I talked about. So how to do some sort of modeling of microbial communities. Just a quick repetition that, of course, you can choose from many different types of modeling systems, from cellular automata to more detailed complex systems with complex metabolism. You can differentiate time scales. From this bunch of different models, I will demonstrate mainly the two most commonly used approaches in microbial community modeling. One is this cellular automata model, which is, of course, as I mentioned, and you probably need to know, it's an individual-based model. And the other one is this cellular population model, in which one cell is a very small two-dimensional, three-dimensional space in which biomass can grow and then propagate into the neighboring cells. And then these two pics are also, I picked these also because one is more like a rule-based modeling approach, so in which you set up certain simple rules that you attribute the agents with. So their detectors, their decision functions or decision rules, and then their responses or their actions or their behaviors, and then also their entire states, right? And the other approach is the equation-based, in which you have a complex structure, maybe a metabolic network of a cell, and then you have to solve a lot of equations and optimize different solutions for something, for example, for biomass. So one will be NetLogo, the other will be the Comets toolbox. So these are the two models I will show you. So the first one is NetLogo, as I already mentioned in the presentation. It's a freely available agent-based modeling platform in which agents can be represented, many types of agents can be represented in a continuous or in a discrete space. It's based on Java or Scala, and then it's very popular, so you can see many different type of models can be done, from this type of segregation model to some kind of rock, paper, scissors type of dynamics, to some aggregation, fractal growth. So I just show you, if you don't know, that there is a version which can be downloaded and installed on your desktop, but you can also run NetLogo in your browser. So if you see how it's done, yeah, so you come here, and then there is a randomly chosen simulation from the library that you can choose, or you can also, yeah, you can also browse the library. So for example, this one I like. So this is kind of simulates the spread of fire. So you can manipulate. You can have also always some manipulating bars or manipulators here, which you can use to set the main parameters. Here is the density of bushes or trees. And then if you hit go, then you simulate some fire starting from the left-hand side. So of course you can say that, well, how fire goes in this setup, and then also say, well, we have a very dense population, and then you can see how fire spreads through the whole And then, yeah, basically with this, you can simulate some sort of packaging effects and also maybe percolation effects. And then, yeah, here you can see the code and some model information, right? What it does, how it can be used. So that about NetLogo. But coming back to the to our presentation, I chose one model which models microbial biofilm growth. So it's called Bacterial Persistence in biofilms. In this model, there are many different things that you can set up, but first we just want to see how cells from an initial founder cells grow on the surface and how the layer of the biofilm gets thicker. So there is the biofilm, which is attached to the surface. And then there's some boundary layer in which the biofilm can grow until, and then you have some diffusion of substrates that the microbes use, and that diffuses in the other direction. So now I come, this is the standalone file that I can use. Now I use the desktop version of NetLogo. So here you can see the same thing. So this is how it's set up, but just to have a screen smaller size, here I set so I have a smaller window that is the simulation space. So again, you have some information. You can see the code here. So you can see the main parameters here, turtles, which are the names for the agents. Yeah, you can see the diffusion procedures or module is set here in this particular code. And then so these are the basic parameters. What we will do now is say that we set maybe 20 cells of initial cells, and then we set up, yeah, switch these up. So we set our simulation. So we randomly set the cells here. We color the lineages, so every cell gets a different color, and then we start our simulation by hitting the go button. And yes, you see as time passes, cells start to divide, and then obviously they will fill the empty spaces. You can also see that in real time you can follow the number of cells and the thickness of the layer. So if you have time to wait, then you can see how it grows. I will stop my simulation and go back to the presentation. And just to show you how, so I did a long simulation here, and then I just made a GIF out of it. So you can see how this biofilm growth looks like if you have enough time and you can wait. So this is very basic setting, and this code was actually developed for a paper by Carvalho and colleagues, in which they modeled two types of microbes. One is sensitive to the antibiotic treatment, so it can die if antibiotic hits it. And there are the persisters, so-called. So these are ones that have a lower growth rates, but they are much less likely to die from an antibiotic shock. And then, of course, these can all go to a dead state. So the model looks like this. So these are the three types of cells. We have the green, the sensitive, the red ones will be the stress tolerant types. we have the same medium, there is some diffusion, and we will use this, going back, restore parameters. Now you can see that it's yeah, you can adjust the treatment here. So it starts maybe after 1 hour of bacterial growth. You can set the duration of the treatment and the recovery time. So it means that if you set the simulations, then they start to grow. So here time is measured like this. Yeah, I don't know if we should wait. We will wait a little bit. Goes quite quickly. So you see, as cells divide, there are a couple of rules in this paper. They said stochastic random switch, and this is strategy 2. So now we apply this strategy. So that it means that cells can randomly switch to this persistor strategy, or you can have an antibiotic or substrate dependent switch, and then they grow normally, and then at time 1 hour, we shock the whole biofilm with an antibiotic treatment. So here now you see that it's shocked, and then we have some dead cells. Now dead cells, from dead cells, when the antibiotic treatment is done and it dissipates, then these dead cells go back to reproductive state. So this is how the simulation looks like again. So you have a growing biofilm, it gets big, and now we shock them with antibiotic. So all the sensitives basically die out, but the persisters, they survive. And once the antibiotic treatment is gone, then you kind of go back to normal growth. So this is motivated by these antibiotic shocks, right? Biofilms and such treatments. So I forgot to mention, if anyone has any questions, you can just ask me. I don't see the chat windows necessarily, so I don't know where is it, but maybe if you just come in voice, then if something is not clear, just let me know. So this was the rule-based model, right? So it's very simple. You can do many fun things about with that. And then we come to this constraint-based modeling, which is already discussed was discussed in the morning sessions. And in particular, we will use this Cobra toolbox, in which you have an object that is filled up with a model. The model is the organism, so it's basically the species that has the genes and metabolites and reactions that happen in its core metabolism. And then you set up the reactions and the metabolites that happen in this arena or in this simulation, the genes that control these, and then you set up the solution. So this is the environment, right? So you have the genes that determine what reactions are catalyzed, and these catalytic modifications modifies the metabolite in the environment. So that's how the model looks like. And the other toolbox, which is very useful, as I mentioned earlier, is this Comets toolbox, which is this computational method for microbial ecosystem modeling, which time and space models. So you can, of course, model time series changes, but you can do it in a well-mixed media and in a spatial media. So it was implemented in Java by this, you can find it in this link. So it's developed and maintained by the members of this Segre lab. So there was an old version, but now they updated it to a bit more realistic. And I think I showed you, but maybe I will also, so yeah. So you see that the new version now contains, for example, evolution, and you can have spatially varying diffusion and stuff like that. Whereas earlier version, it was version 1 was a bit simpler. Yeah, and just to repeat what I told you in the morning, so you can have continuous or well-mixed cultures. You can have a spatial culture like a Petri dish. And then you have basically the metabolic model of the organism. You have the composition of the substrates of the environment, and then you can also define extracellular enzymes. And then you have the model, the layout, and the parameters which determine the simulation, right? The model is the organisms, and these are the environmental parameters. And then you can put them together using this core engine of the Comets, which uses Gurobi optimization. And then you can run it in its own general user interface, or you can run it in command line, or you can run it you can have a Python toolbox, install it as a Python toolbox, or load it, or run it in Matlab. And what we are going to do is I'm going to show you a couple of examples using the GUI and the Python and the Matlab toolboxes, right? So I showed this already. So this is how the workflows look like. So you can see these things all from the groups lab web page, or from this publication, which includes many of these examples, which I will show you. So you can find the list and the descriptions there. So first, I am going to show you how to do a simple model in using the Comets workflow with its own user interface. It's going to be a simple E. coli model. So it's going to be anaerobic fermentation on a minimal media in a virtual test tube, and it's going to be the only carbon source will be some glucose that we put in the beginning, and then see how the initial biomass of bacteria grow, and of course the glucose will be consumed, and we will see some waste products accumulating. Okay, so now I come to Matlab. So I don't know if you're trying this. I already initiated the Cobra toolbox with this command, initiate Cobra toolbox in Matlab. So this, you can see that it uploaded all its different components. And now what we're going to do is that we're going to run this E. coli model, or construct the model itself. So first, we have this E. coli core XML, which is the E. coli model. It looks like this. So here you see the different components. So for example, this is the stereoarithmetic matrix, right? So you see the entries, the pluses and the minuses, all the reactions. So you see the metabolites here. So these are the important metabolites, lower and upper boundaries of the fluxes, the genes involved, the different the gene names. So you have the names here. So these are the designations, and then you have metabolite, some reactions, right? So this is, you can see different reactions, the enzymes and everything here. So how does this look like? So this is what you see if you load this E. coli core model. So it's loaded from this big models database, which is a couple of 10, maybe I think 70 different organisms uploaded, and then different their most core metabolisms and the relevant information. So you have the list of metabolites here, right? So these are designations and the names and the models, and then if you load, for example, acetate here, you see that it's involved in the E. coli core model, and then you see it in the metabolic map that is included in this E. coli core model. Then you have the genes here, their sequence and what are they doing and the reactions, right? So say the different reactions and enzymes, and also the pathways that are the parts of the metabolic pathways that are relevant here. So we do this, you see here. We have the set the core model here. Then we set, this will be the output file that will be used for the Comet simulations. We set the word. So the word is now one times one size of a cell. So we simulate one single grid cell that will have all the things there. We set the media, so the glucose will be in limiting concentration. So this is the initial concentration of glucose that we put in our test tube. And then we set all other relevant media that is needed here in unlimiting concentration, right? So 1000 means that it's unlimiting, or 0 that it's not present. This is the initial population size that we put in our test tube. And then here we write this file out. So now we created this batch layout file. Which is now, we copied to this Comet simulation. Now we start our Comets user interface. So in the beginning, it asks us to load the layout file, which we just generated using Matlab, but there are several other ways how you can do it, basically. So you come to this directory. So here's the Comets batch layout that we just made. We load it. And then we also need to load some parameters here. So these are the external parameters, like you see the number of cycles, how long we run the simulation, what is the output file name, or the, the steps for the numerical integrator and stuff like that. So other parameters, you can change these, and then now we have to load these parameters. The global parameters. We set up our simulation. Basically, you see that everything is set up. Here you can see our media. And then run the simulation. So I don't know why it's not following, but you see our simulation is running here. And then in the end, it will create an output file. And then if we plot this output file, we should see this. So our numerical simulation just did this. So we started with a low initial density of E. coli, it grew. And then once the glucose was depleted, it stagnated there. And then, of course, there are these waste products that came out of this metabolism that got accumulated in our test tube. So this is how basically you set up a simple simulation. Do you have any questions? If not, then we move to our next exercise, which is going to be a Matlab example, also, of course, using Comets. So Matlab is this matrix laboratory. It's a very good programming language, but unfortunately, it's also not easy to get. So it's not necessarily free. I'm now using a trial version of it. But anyway, it's very useful. As you saw, for example, we did the Comets layout with Matlab, but now we will use the model, this mixed culture model, in which we basically somehow use the information from this article. So it's they did pairwise experiments for cross-feeding, and they put together these 14 strains. Yeah, so I want to okay, so this is how it looks like. So here also we find the metabolic model. So I'm not going to go through all the details as before, just show you quickly how it's done. So you set up load the model, create the Comets layout, load the medium that is defined separately here. So this is the, yeah. So these files are in this folder, and then you set, for example, these nutrients in limiting amounts. You set the initial biomass. You define the cycles here, like the different parameters. Now I set it to 1 because it runs for quite a long time. Yeah. So let's run this. I hope it's going to run. So we see that it's kind of started running. So it's creating its own directory. Here, putting all the different logs and biomass information here. Yeah, and you see here already included in the end of the simulations or after the simulations some plots. So this is the for 10 iterations. You see the plots of the different species, and then you see that the two limiting substrates that we put there, glucose and alanine, are being depleted. So basically, if you run the simulation for, I don't know, 1,000 cycles, you see this, that there are some species that outgrow the others. This Methylobacterium, I think, this extorquens, yeah, so this grows really fast, you see, and the other ones, and then you see that the media gets depleted, and then it goes down. I mean, growth goes down as once the main resource is depleted. But we can do one more thing with the help of this software, and I already did it, as you see there, but I just show you. So there is, these are the plotting modules or code, and here, what you can say that you look for the different metabolites that are secreted, absorbed, and exchanged. Basically here. Yeah, so secreted and absorbed metabolites are the highest flux of metabolites that appeared during the simulation. And then the excreted one are the exchange metabolites. So how it looks like. Yeah, if you come back, for example, absorbed metabolites, you see the 14 columns are the 14 species, and then these are the metabolites by in the different rows. And you see, for example, here, right? So these are the main absorbed metabolites. But what's more interesting is this exchange table, which says who secreted, who consumed, and what metabolite. Sorry. You come here, right? So you see there are this is the list of metabolites that we sorted out from all the metabolites and all different reactions. So we say, for example, species here, 2 here produced metabolite 17, which was taken up by species 5. And then we come to others. Model names. So species 2 is the E. coli, and it is picked up by species 5. So by this Methylobacterium extorquens. So you can see, and metabolite 17 is the acetate, actually, if we look up in the table. So what we can also do is that we sort out these metabolites, and basically here you see the results. So you see that actually this is the metabolite that was produced and then produced by one species, and then or few species, and then consumed by others, right? And this acetate is because of this reaction of the E. coli. But there are other species that actually in this test tube consume this metabolite. So I just quickly want to show you another example. This time is flying. So, so this example is set here, but maybe I will not run it. So it's going to be this day and night cycle of this Prochlorococcus, which is in the ocean, right? And it uses light for photosynthesis, and then we can actually model some sort of this sort of cyclic environmental conditions also using this software. So if we run this simulation, it's going to run quicker. We can wait. We will have 2 cycles. And the results here. So you see that the biomass grows during the light cycle, which we have we defined increasing and then decaying amounts of light hitting the organisms, and then it stagnates during the dark cycle, and then it grows, and then you can see also the different metabolites involved. So any questions at this point?
I have a short question now. That were these previous examples well-mixed system or not?
Yeah. These were well-mixed system. Yeah, as I said in the beginning, the first one, so it was one grid cell, which is a small biomass of well-mixed medium.
Yeah.
Right. So it's and that's why we actually come to this next exercise of ours. And now we will use Python just to show you how it works with Python. So if you don't know Python, it's also a very popular programming language, and many packages and modules and libraries are developed, and you can load them. For example, Comets is the Python toolbox of the Comets software, and then you can use different means. You can use Jupyter, which is the browser-based version running of Python, or you can have Spider. Yeah. Yeah, I think we don't have so much time, so maybe I will just skip this one. Okay, so come to here. And as I said, Comets makes spatially explicit models in this way. So you have a grid cell, and one grid is basically a little well-mixed system with a little biomass. And then propagation or dispersal or diffusion is going like on a normal grid cell, so into the neighboring areas. And then you can set the diffusion constants, right? So you can basically do different growths. And the first one I would like to show you is the spatial explicit model in its own general user interface, so with the Comets. Just to show that layout. So now we loaded all the parameters and everything, and then we have one small biomass set in the middle cell, and then you can start the simulations. We unclick this so that it goes until we stop it, and it starts simulating, and you see that basically there is some growth going on. And then you can do different fun things. For example, you can manually manipulate the spatial setup and then start running simulation from this stage, or you can add cells manually and stuff like that. So that's a nice software, but of course it's not very easy to run simulations with it. So now we are going to So again, I run a longer simulation just to show you that you can define different colony growths, right? So this is also based on some biomass. You can set different biomasses and local concentrations and diffusions, and then you can set if the colony growth should be, somehow this coral shape or more circular colony growth. So you can simulate all sorts of growths like this. So now coming back to Python, now we will do the spatially explicit examples. So we can call virtual Petri dish experiments with this Python. So we use the CobraPy, which uses Cobra, and CometsPy, which is the interface to run the Comet simulations, right? So this is how you start, for example, Anaconda Navigator you can use, and then you start Jupyter or Spider notebook or anything that you want. I already had here Jupyter, so I just load the written script here. So you here start importing the Cobra and then the CometsPy, the necessary packages that you need for this simulation, right? So you run the cell, clicking on run or the control, enter, you run the cell, and then you model the E. coli Cobra. So this is the E. coli model, which is included in the Cobra package. So we don't have to download and load the model itself. It's already there. We just have to load to our simulations. Then we can define the initial density, and the size of the thing and so many things. So here, what I do is that in the middle of the field, I put this biomass, this amount of biomass of initial population, right? So this will be the grid size, and then this is this is the diffusion and other parameters. And these are the metabolites. Again, glucose will be in limiting amounts, and the others, water, will be in unlimited amount. And then with this command, we can see that actually what is there, right? So we just check. Here we set the other parameters, running cycles. I'm sorry. And then here we just run the simulation. So it started running, you see. Now we only do 10 cycles. And here we use the plot, which plots the results. So this is after how 10 cycles our colony looks like. And then this is how the resource looks like. So it's depleted when the colony grew. and some growth rates. So basically, we can experiment with this different amounts of resource or different initial biomass we can set, and then we do this. But then we can also run another experiment. So I made a couple of experiments, basically. So, yeah, I just quickly show them. My time is almost up, I think. But what we do here, because it's also I think educational, what we do here is basically we do the same thing, right? So we load all the parameters in. I just put it in one field, but we put three initial populations on our Petri dish at three different locations, one in middle amount of biomass, one small and one high amount of initial biomass we set there, right? And then we run the simulation, which is now running. Maybe I don't wait for the results, but I show you how this looks like. So now we set the three initial biomasses here, here, and here in these locations from far one away from each other. Of course, the larger biomass grew the largest colony after a fixed amount of 1,000 generation cycles, right? But what's also interesting, as you can see, that the glucose, and of course the glucose was diffusing, had a small diffusion rate. So what you could see is that, of course, the biggest colony consumed the most amount of glucose, also sucking away from the environment. And then the growth rates of the colonies are higher towards facing the higher resource concentration. So, for example, you see here that this poor guy is growing more towards this region because on the other side, this big colony is sucking away the resource. So it has very limited resource here, but very high resource concentration in the other direction. So this is how it's growing. This is the growing direction. Yeah, maybe there are two other experiments. Maybe I just show the results, right? So because these are the same experiments, I just, I was just playing around. So what you can do is that you, for example, define two different diffusion constants for splitting your Petri dish and then start growing colony. And then, yeah, this is just a simulation of this. So you see that on the areas where the diffusion constant is higher, more resource gets into the colonies, right, from the surroundings, so they grow bigger. And of course, here we set the same amount of biomass, no locations, just the diffusion is different. And you can also see these growth patterns when the colonies are close to each other. They grow asymmetrical shapes so that the growth rate is higher. Once they kind of consume the resource in between them, they start going into the other directions. Yeah, and here just you see the different plots. So these are the biomass, right? The growth rates, the acetate, which is accumulating, and the glucose, which is consumed. So you see the different maps here. Yeah, and the final experiment I just quickly show you, so you can also set different settings, for example, by 4 different diffusion parameters. And what's interesting to see, of course, that the colonies at the edges grow always grow more in the direction of higher diffusion rates. And you can also define different morphologies, different growth rules using Comets. And yeah, here are the references of the papers.